Flow cytometry is particularly valuable because, first of all, it's been around for a long time, so it's almost like a gold standard technology.
We use flow cytometry to quantify and characterize immune responses in patients in multiple diseases with as many markers as we possibly can include.
Current innovations within that field mean that we are now able to get a much deeper understanding of the immune system and the immune response to disease states, and that really helps with the development of these personalized medicine and treatments.
To understand disease at the individual level, researchers need tools that reveal detailed information about individual cells. This is especially important for identifying and characterizing diverse immune cell types within complex samples.
Flow cytometry is a high-throughput technique, so it helps us to gather a lot of information quite rapidly. And also because of the immunophenotyping capabilities, you're able to get a quite complete view of what is going on in the disease state.
Well, flow cytometry is basically a tool that allows us to look at protein expression on cells and allows us to characterize the cells that are involved in an immune response.
Well, the immune system is a very complex system or network of cells that are distributed throughout our entire body and that can easily be accessed through our blood as these cells move from organ to organ through the blood. And the immune system is incredibly heterogeneous. So the diversity of cell types is really overwhelming.
That complexity is exactly what flow cytometry is designed to capture.
When we measure all of that information cell by cell, we're able to very precisely and very finely dissect the immune compartment or the sample compartment to understand exactly what types of cells are present and to group them in different ways and understand which are important in an immune response.
This cellular level resolution can be applied across diverse sample types and proves valuable in both research and patient care.
Those populations can come from samples like blood samples, bone marrow samples, and also we can use them to understand more about tumor samples in disease states.
That's particularly important for precision medicine because that allows us to identify the exact type of cell or group of cells that will predict whether a person responds to a therapy or doesn't respond to a therapy, has toxicity from a disease or a drug, or doesn't have this toxicity.
Precision medicine really is about developing therapeutics and treatments that are tailored to individual patients. So think about developing a treatment that is going to target an individual's unique tumor and really just being able to make sure we're treating each individual patient with something that's going to meet their individual needs.
To do this efficiently, scientists use technologies like flow cytometry to identify what makes each person different. But targeting the tumor is only part of the process. Understanding how the immune system responds is just as important.
Tapping on immunological information, it's becoming probably the next frontier. Also linked to the fact that there are so many immunotherapies being developed.
There's so many drugs that modulate immune responses. We actually want to show that they, in reality, in everyone modulate immune responses in the same way. And all of this work that we're doing sort of at the cohort level on groups of people, we hope will translate to individuals. And that's really the idea of precision medicine, to identify biomarkers that classify progressively smaller and smaller subsets of patients so that we can understand the differences between them, and then to make the leap to translate those into using them individually on patients. And that's where precision medicine will really come into action with flow cytometry.
So how exactly does flow cytometry provide this critical information?
Flow cytometry is a technique for analyzing cells in a fluidic stream moving rapidly through lasers. So these cells get labeled with a fluorescently conjugated antibody. These antibodies help us to develop a deeper understanding of the immunophenotype of the cell populations that we're analyzing.
What we're doing in flow cytometry is basically characterizing the cells that are present in those sample types. And the way we characterize them is according to their protein expression, typically.
This process involves 4 elements, which are the cells, the fluidics, the optical system, and the detection system.
On the first hand, you have the cells and how the cells are treated so that you can measure them. Typically stained or incubated with antibodies. Antibodies will bind to the specific targets that you want to measure, and these antibodies are labeled with fluorescent molecules, with fluorochromes, and there are many different types of fluorochromes with multiple spectral properties, and thanks to this we can combine lots of antibodies in the same experiment.
Those fluorescently conjugated antibodies can then be used to identify proteins that are expressed associated with a particular cell, and then we can make those measurements on a cell-by-cell basis for every cell in a sample, kind of maximizing the information that we're getting from the cells that are in that sample.
Importantly, the cells with conjugated antibodies are suspended individually in fluid so they can be measured one at a time.
As they are placed inside the instrument, there is a needle that will suck the content of the tube containing the cell suspension in the fluidic system. And thanks to a hydrodynamic flow of a stream that will push the cells in the middle of the stream, the cells will be sort of positioned in a way in which you can measure one at a time when they arrive to an interrogation point. At this interrogation point, a laser is focused precisely at the center of the stream, so that when a cell passes by, the laser will excite the fluorochromes attached to the antibodies present on the surface of the cell or inside the cell.
When excited by a laser, these fluorescent molecules emit light with distinct spectral properties, which is then collected by detectors through fiber optics for analysis.
And these detectors vary from vendors. Some use photomultipliers, some use avalanche photodiodes or CCD cameras. The important thing is that they amplify the signal and then they bring it to a computer that ultimately will quantify how much of that antibody was present per cell.
In the end, a flow cytometry experiment produces a table where each row represents a single cell, often millions in total, and each column corresponds to a measured parameter. Each parameter in flow cytometry represents a fluorochrome-linked antibody, and depending on the method used, data must be transformed and corrected to reveal the expression patterns of molecules in each cell.
So with a single test, maybe we can measure 40 or 50 different proteins on the surface of cells, one cell at a time. And this gives us an enormous power to classify all these cells into their most detailed phenotype.
And so you can imagine that if we measure multiple proteins at once, really what we're doing is getting different combinations of proteins that are expressed or not expressed by particular cells, and then we're able to identify or characterize or name those cell types on the basis of those combinations of proteins.
Flow cytometry's high throughput and multi-parametric analysis capabilities make it ideal for numerous applications.
Flow cytometry can be used for so many things, for monitoring cell cultures, investigating cell division, or signaling cascades, or the immunometabolism, even morphological changes in cells with the help of imaging. So there are many, many different applications to flow cytometry. But in particular, in my lab, we are mostly interested in performing immune monitoring on patients, mostly cancer and also neurological patients.
In one recent study, Dr. Garcia Vallejo and his team combined flow cytometry with single-cell RNA sequencing and protein profiling to uncover how specific immunosuppressive macrophages promote tumor growth in metastatic colorectal cancer. This kind of integrative analysis demonstrates how flow cytometry can be used to understand disease. In addition to this, flow cytometry plays a vital role in diagnosing and tracking serious diseases.
So, for example, in HIV disease, flow cytometry is used to count the CD4 T cells that are present in somebody's body that tell you whether their disease is progressing to AIDS or not, that tell you whether the therapies that are given are working or not. The second area where flow cytometry is particularly useful in clinical medicine is in staging leukemias and lymphomas. to understand what type of leukemia a patient has, or to understand how far along their lymphoma is, the staging of the lymphoma.
This works by identifying specific proteins found on the surface of circulating tumor cells and providing critical information for accurate diagnosis and treatment.
And the third way that it's used in kind of a quasi-clinic adjacent setting is to really understand the cells that are involved in an immune response or in the response to a particular drug that targets the immune system.
These applications have solidified flow cytometry as a key component of many precision medicine workflows. Because flow cytometry is highly versatile and compatible with a wide range of sample types and staining panels, it can also fit into many different workflows and allow for even deeper analysis of important tissues.
Flow cytometry can be adapted so that we can use it to sort out and purify particular cell types that may be of unique interest to a researcher. And when we use that fluorescence activated cell sorting, that sorting based flow cytometry, we can actually take those purified cells and then look at them in other ways. So instead of just looking at the proteins that they express, we can look for epigenetic changes that they might have. We can look for changes in transcript expression. We can look for DNA characteristics that are just inherent to a patient's genetic makeup. And we can also do metabolomics, where we're looking at the metabolic activity of the cells. And so flow cytometry really becomes a critical central point for other technologies that could inform precision medicine as well.
Although flow cytometry requires cells to be in suspension, this isn't limited to blood. Solid tissues, such as tumors, can also be broken down and analyzed at the single-cell level.
One of the approaches that people are using, in particular in cancer immunology, is analyzing the tissues, cancer tissues, for example, in suspension, so that you can very easily characterize the heterogeneity of the tissue, and then select the number of markers and then measure that on tissue slides, so that then you can actually connect all the different frequencies that you found in flow with the spatial relationships that are present in the tissue.
Combining flow cytometry with these complementary technologies allows scientists to link function, identity, and spatial organization, which increases the insights into important tissue samples. Flow cytometry has grown from a basic analysis tool to a sophisticated platform capable of high-throughput cellular analysis.
There have been a lot of changes in flow cytometry technology over the last decade to 15 years that have been really remarkable for the impact they've had in the field and for the value that they've offered. Some of those advancements include new reagents, new fluorescent dyes that help us detect more proteins at once and that help make the detection of those proteins really clean and discrete from background noise. So advances in reagents have certainly been important.
I think one of the recent innovations that's really had a huge impact across flow cytometry, not just in precision medicine, has been the emergence of spectral flow cytometry.
For many years, there were very small innovations that were helping us move from 5 to 7, maybe 9, 11 markers at a time. It took a long time to get there. All of a sudden, spectral flow cytometry has helped us move from 15, 20 into 40 colors very quickly. That has been really transformative because it is still very affordable as compared to conventional flow cytometries.
Spectral flow cytometry differs from traditional technologies because it captures the full fluorescence emission spectrum rather than just a narrow wavelength band.
And that allows us to distinguish different dyes more clearly and add more markers to our panels so that we can look at more things associated with cells.
This has enabled people to really expand on the size of panels that they're running in flow cytometry, get a much more granular understanding of the populations they're looking at. And of course, with it being a high throughput process, you can get to statistically significant information quite quickly.
I think that this has had an incredible impact in the field of immunology, and it's going to continue having a very, very important impact in the coming years. And I think the number of publications during COVID addressing the COVID-related immune response was extremely overwhelming and definitely facilitated by the availability of these kind of technologies.
When we think about precision medicine or even any of our disease studies where we might be working with rare samples, small samples, and patient samples where we have one shot to get the information we need, being able to do big panels means we can ask multiple questions in one sample. We don't have to break panels up into smaller sizes. We can do it all in one go. And I think that is a real value that spectral and high parameter flow cytometry brings.
On the hardware front, there's also new instrumentation that basically treats the cells very gently so that the cells aren't essentially trashed as they're going through the system. And because of that gentleness, you can actually sort these cells and be confident that their function isn't an artifact of them going through a machine or some experimental process, but is hopefully more akin to what is happening in the body. And then the third advancement is one that I'm particularly excited about, which involves dynamic measurements, where we make repeated measurements of the same cells in a sort of time-lapse fashion. We call that time-lapse flow cytometry, and that's emerging right now also.
Traditional assays capture just a moment in time, but with time-lapse flow cytometry, researchers can analyze cell samples at multiple time points to track dynamic changes in cell behavior or marker expression. This approach reveals how cells respond over time, and it offers insights into processes like drug response, immune activation, and cell differentiation. As flow cytometry technology has grown, so has the scale and complexity of the data it produces. This has introduced several difficulties into the analysis process.
There are a number of challenges to analyzing data in this space. The first is that there's just so much information.
For me, as a director of a microscopy and cytometry core facility, this is a problem that I face every day. On one side, actually, to have the sufficient infrastructure to be able to store this data so that people can analyze it in the future, and also to provide the tools to the people doing research in my core facility to be able to extract the information they need.
If you think about a cell expressing a marker or not expressing a marker or just ignoring that marker, then you have three conditions basically that you're looking at. And if you measure 20 different proteins, it's 3 to the 20th different cell types that are conceivably within that sample. And to measure so many cells and quantify them and track them and to graph them can be quite difficult and requires some elegant thinking about how we show these types of data sets.
Standards for data formatting and sharing are also becoming more important as researchers seek to merge data sets and enable cross-study analysis.
Another challenge that we face in this space is that we have a tendency to want to cluster cell populations together so that we can see them clearly on a graphic and show them and share that information between scientists.
Many current tools lack built-in statistics that show variation across patients or samples, which makes it harder to interpret results and assess reliability.
We can, frankly, until the cows come home, identify different cell types that are important in a disease setting or to a therapeutic response. But if we can't contextualize that information so that we understand that this population is important and was reported in 1992 by this group also in this other disease setting, if we can't connect that information to literature and to our understanding of biomedical research, that's a huge problem. And that's really one of the main problems that we're facing now.
Researchers like Dr. Chattopadhyay are addressing this challenge by building comprehensive tools, such as TerraFlow, a platform designed to match newly identified cell types with existing literature.
The next frontier is going to be how do we integrate multiple data sets together, and also with other omics, maybe with proteomics and with genomics as well.
In terms of informatics, we're able now to wrangle massive data sets where we're getting millions of cells and hundreds of parameters worth of information, and we're combining those into single data sets and being able to analyze that successfully with new tools.
I'm very happy to see that in our community, in flow cytometry, people really want to do this as well, and more people are doing this, which means that eventually there will be more than enough data sets to actually perform reanalysis and maybe even meta-analysis of different studies altogether. And so that's going to be extremely helpful, and I have very high hopes for what this could mean for personalized medicine in the future.
These continual advances, integration with other technologies, and improvements in analytics make flow cytometry a key tool for deeper insights into human health. With each development, it helps us better understand disease and design more effective treatments for patients. Stay tuned for our final segment, where we'll cover the current state and future of precision medicine.